Genome transplantation was invented by Carol Lartigue in 2006 after 2 brutal years of desperately hard work. And this was critical to our being able, to the JCVI, being able to put out these 2 papers. SIN1 in 2010, where we made, in essence, cloned an entire Mycoplasma mycoides genome with a few modifications, some watermarks, and then the minimal cell in 2016. And if you followed the scientific literature, so with both of these papers, but especially SIN1 in 2010, there was predictions from many in science that we were entering a new era where people would design and build genomes, millions of them, and then transplant them and boot cells up. And this would just usher in a remarkable change in synthetic biology and what we can do with life. And so here we are, 12 years after SIN 1.0, 15 years after the development of transplantation. And at this point, there have been two other synthetic cells, both of them E. coli. Jason Chen has made a E. coli with a synthetic genome, and George Church has almost finished making a E. coli with a synthetic genome. And they couldn't use genome transplantation. Instead, they had to build a synthetic genome in a lot of pieces and then install each new piece, in essence, by recombining or swapping that out with the corresponding piece that's inside the cell, that they're inside the native E. coli. And so for, I think, the Chen group, it was like 55 steps. And it took them a very long time to do this. Now, right now, SDI, or now Codex DNA, Dan Gibson's organization, at one point were advertising that they would be willing to synthesize a 2 megabase genome for someone, and I suspect they still would, to build that genome in yeast. We've even seen that it is possible to clone much bigger yeast artificial chromosomes, if you will, in yeast. There is a Chinese group was able to make a yeast strain where all of the chromosomes have been assembled into a single chromosome. So it might be readily possible to make yeast artificial chromosomes to clone any bacterial genome into yeast. But right now, until genome transplantation is made to work in non-mycoplasma cells, it's not available. And so what's left is this piecemeal reconstruction of genomes, which is a very long, slow process and very difficult. So it would be fantastic if we could do this in general. Now, for SIN1 and SIN3, we had to develop 3 basic technologies. Genome synthesis from oligonucleotides, which was an expansion of methods that had already been demonstrated for DNA synthesis. We just developed new methods, and it was at a vastly larger scale than had previously been done. We had to learn how to clone bacterial genomes, park them, if you will, inside a yeast cell so that you could get enough genomes to then do this transplantation reaction. And these were the 3 technologies. Now, Carol's transplantation technology, it's sort of like this. This is how we envision it, our naive model, if you will. We have a genome, synthetic or not, that has an antibiotic resistance marker. And we're going to treat recipient cells with polyethylene glycol and calcium ions, so that somehow that genome enters a cell, probably like a conventional chemical transformation of E. coli. We'll take a cell that is transiently diploid and put it under growth conditions so that you will get daughter cells with different genomes. These then can be treated with tetracycline so that the the cell that had the native genome, in our case, a capricolum genome, will not grow, and the cell that has the added genome will grow. Now, we can also, in a method that I desperately need to get the paper finished on, we can treat the recipient cell with mitomycin C, which is an anti-cancer drug that cross-links, covalently cross-links nearby G residues, either on the same strand or on the opposite strand. So in essence, we destroy the resident genome, and then we can put the new genome in, and we can do this without any antibiotic selection if we want to. So this is one of the things that we can do now with transplantation. As I go through this, I'm going to give you some things that we have learned about transplantation that may guide us as we think about trying to build transplantation for non-mycoplasma bacteria. So a summer rotation or intern student, and suddenly his name escapes me. I should remember this, but I don't. This was 12 years ago. He's now a physician not doing science. But we loaded recipient cells onto these Nicodens gradients and that were step gradients, and then isolated the cells from different bands in these gradients. And we found, for instance, that this band gave very few transplants, and this band gave a whole lot of transplants, suggesting that the recipient cells are a population with different characteristics and different capacities for transplantation. So there may be a better way to prepare recipient cells than we've currently got going. Now, transplantation, as we have said several times today, works within the mycoides clade of mycoplasmas. We tried desperately hard, Naysira Assad Garcia, after Carol developed the transplantation technique, we tried for years to transplant genitalium into pneumoniae or genitalium into genitalium, because these two organisms are extremely close phylogenetically. Every gene, or ostensibly every gene that is in genitalium has an ortholog in pneumoniae. We never got it to work, although we did get cells in genitalium to genitalium, two strains of genitalium, where that had two antibiotic resistance markers. We got large pieces of DNA, maybe more than half a genome to go in, but it was a chimeric genome of both strains. So that never worked, and this was really frustrating, and eventually we just gave up. We also, Bogomil Karas, who spoke earlier, tried transplanting Haemophilus influenza in an intra-species transplant as opposed to an interspecies transplant. As I said, Naysira did genitalium. She also tried to transplant Mycoplasma gallisepticum, again, intraspecies transplants. Now, eventually, Naysira did experiments where we learned that the calcium treatment of genitalium or pneumoniae recipient cells destroyed the donor genomes before transplantation ever had a chance. So calcium activated the DNA on the surface of the genitalium. Now, here's an example where 2 years in the lab can save you 2 hours in the laboratory. So in 1993, Chris Minion published a paper where he said all Mycoplasma species contain nuclease activity, but Mycoplasma capricolum was unusual because its activity was dependent on magnesium and was inhibited by calcium. If we had known that, that would have saved us a vast amount of time. Now, Carol's group also showed that the farther the phylogenetic distance between the donor and the recipient cell, the less efficient the transplantation process. So, Leachii to capricolum works better than Mycoplasma mycoides subspecies capri to capricolum, or mycoides, putrefaciens, florum, etc. Another thing that Carol showed is that while they could transplant Mycoplasma mycoides isolated from a bacterium into capricolum, they could not transplant a yeast clone of Mycoplasma mycoides into capricolum. Now that suggests to me that if we sequence a genome coming out of yeast, or a mycoides genome coming out of yeast, and look for modifications of the yeast genome, we look at the modification, we may find that there are modifications made by yeast that are problematic for Mycoplasma mycoides. It could be we're activating some kind of restriction system. I don't know. That's something that we're going to have to think about. But these are things that are important. It says that if you're going interspecies, the donor and recipient have to be very close. Now, we also did design of experiments analysis of the importance of factors in genome transplantation. And so you see some things really didn't matter and some things were really important. Capricolum recipient cell growth temperature, incubation time on ice, the experimenter doing the transplantation. So there was a point when I sat in a hood with Carol and did transplantation. We used the same reagents. I had my pipette, she had hers, and she got like 2,000 colonies and I got 20. A, I'm that bad, but B, it shows that this is a difficult technique that is really a little bit like art. To date, no one except Li Ji Sun in my group, to my knowledge, has been able to read the transplantation technique and be able to develop it on her own. Everybody has had to either work with Nasira Assad Garcia or to go to Carol's lab or go and work with Li Ji to get this done. So that's an important thing to consider. So our plan for developing mycoplasma bacterial genome transplantation using non-mycoplasma, we're going to do it with two organisms. We're going to do intraspecies transplants with mycoplasma, with Haemophilus influenza strain RD. Now that'll be the donor. That was the strain that Ham Smith used to discover restriction enzymes, and it's a BSL-1 organism, but it's got a 1.8 megabase genome. It's gram negative. The other strain we're going to use, so Carol's working with Bacillus subtilis, which has a huge 3 megabase genome. We're going to work with Streptococcus thermophilus. It's again gram positive, small genome, BSL-1, and even though it says thermophilus, it grows really well at 37 degrees. So this is part of the plan. We're also going to work with Carol, who this will be a collaboration between my group and Carol's group, and I'll explain how James Pelletier and Elizabeth Stralski will be working with us in a moment. Things we're going to have to do, we're going to have to circumvent surface-associated nuclease activity, which is a common thing in bacteria. So one thing we have tried, it wasn't very effective, but there may be better ways to do it, is a trypsin treatment of the cell to degrade any of the nucleases on the surface of the cell. Now, I've said that transplantation may be, it appears to be very similar to a chemical transformation of E. coli. And back before many of you were born, when I was doing transplantations back in the 70s, anyway, there was a point when the rage was, instead of treating your E. coli transformation reactions with calcium chloride, people were using rubidium chloride, and it gave higher efficiency. And this may be because the rubidium is not activating the surface-associated nucleases. This is one of the things we're going to try, but we're going to be trying a lot of things because if Carol has shown anything, conditions really matter for this. So will cell walls in non-mycoplasmas be an issue? We know that large DNAs can be moved into conventional bacteria without getting rid of the cell walls, but if need be, we could think about protoplast. Similarly, will recombination in non-mycoplasmas be an issue? Because mycoplasmas are not that good at recombination, but a lot of other conventional bacteria are. So we may have to go into recipient cells and eliminate recombination activity in order to make transplantation work. But this is a doable thing because most bacteria that people are going to be interested in working in probably have some genetic tools that may enable this. Okay, design of experiments, as I said, showed that the experimenter doing the work is really critical. This is art and science put together. Carol was brilliant at this. Nasira was great at it. Lee Ma, who was in our group at JCVI, people who have done this a lot can do it well. But if you are new to it, your results are likely not to be good. So I was interested in 2012. I approached at a DARPA meeting. I talked with some of the scientists there about could we develop better methods for genome transplantation, something that might involve microfluidics that would make it an automated process. And that led me to Elizabeth Strahalski at NIST, who is a physicist, and James Pelletier, who at the time was a graduate student in physics at MIT. Now he's at the Institute of something in Barcelona, and he spoke to you earlier. But Elizabeth and James, so James would come along with his colleague, Andreas Merschen. They would come to the JCVI and we would work at NIST and at the JCVI trying to develop microfluidics technology that would enable us to eliminate the human factor in at least parts of the genome transplantation process. So this is an image that I've taken from James, that we make these microfluidic devices that have a lump of PDMS that is on top of a cover slip, but this has that has been cast over a microfluidic template, if you will. And so you take this lump of plastic and attach it to a cover slip so that you've got an inlet where you can flow in media with cells. They'll go through this channel and then the media will come out, and some of the cells will flow into these microfluidic chambers. There could be 1,000 of them between inlet and outlet. And so you will move a few cells into these chambers, as you see here. And again, I have graphics I should have thought about. So this is the direction of flow. Cells will flow in. They tend to concentrate at the base of the chamber. Here's some micrographs and that James did early on working with SYN 3.0. Okay, so here's a video micrograph of, we can put the cells in and then the media is there and the cells grow. But the plan for doing this is that we're going to flow so cells containing donor genomes into these chambers. And we will use molecular crowding. So we're going to use proteases to degrade the cell so that we've got a donor genome sitting in the bottom of these chambers. We will use PEG to crowd these things in. And then we're going to, so as you can see here in this idea that you're going to get DNA in the bottoms of the chambers. And James, I could be butchering this, but I'm doing my best. But the idea here is that we're going to move first donor cells, either yeast, initially we were doing this with bacteria, into the chambers, eliminate the protein, everything but the DNA that will be left in these chambers, and then flow in recipient cells. Now, the transplantation process in the process that Carol and others are using, we've estimated that you get maybe one transplant per 100,000 recipient cells. So what we want to do is really just force the recipient cells under transplantation conditions directly in contact with the DNA. So hoping that the DNA would be engulfed by recipient cells and the reaction would work. Too fast. And what we're hoping here is that this will by automating the transplantation process, it will make us able to test conditions at a higher rate for non-mycoplasma bacteria, but we're going to be doing these two processes in parallel. So we're going to be using, for the initial automation development of genome transplantation, we will be working in the mycoides capricolum system that we know and love so well. But we'll also, I'm going to be hiring a postdoc, hopefully soon, that will spend time at JCVI Rockville and also time at Elizabeth Strahalski's lab at NIST. And the idea is that at the JCVI, they're going to be pursuing condition space to try to work it out for Haemophilus and Streptococcus thermophilus. And then at NIST, they will be working on developing the automation, and then we will merge these two. Now, not one part of me thinks that we were so lucky at the JCVI to choose the only organisms in nature that this technique will work on. I think that it will work for lots of things, and it's just a matter of time and experimental persistence before we figure this out. With that, let me see if I can figure out how to get out of this. Hang on. Let's see, Tim, how do I stop sharing? That didn't work.
Hold on, John, I can do it for you.
Thanks. Oh, there we are, back. Anyway, I should have said that this was a work of a whole lot of people, and I forgot to show that slide. Carol, a lot of people on this call, people you haven't seen, and I was a bad presenter in that form, but I've been up all night. So, thoughts? James, Pelletier, Carol, you guys should have some thoughts about this. Carol's going to help us on this project, and we hope to enlist Bogomil as well. And Carol, you seem to be muted.
John, may I ask a question?
Please do.
Do you have any plans to use bacillus for genome transplantation? Because I think it's one of the best understood organisms and it could be used to solve the problems.
Well, the reason I didn't choose bacillus, I wanted the smallest genome I could get. And bacillus at 3 megabases may pose problems that the 1.8 megabase genome won't. I look at it that once we figure out how to transplant, once we can figure out how to transplant a sort of model gram positive and a model gram negative, that the same lessons should apply to bacillus. And so my thought always was that the second organism after Strep thermophilus would have been bacillus, or coli in the case of a gram negative.
Okay, very good.
And again, just a second, Carol. Okay. I think that what we're going to be doing is intraspecies transplantation. No, I get it confused. Intraspecies transplantation. So we would go from one bacillus strain into another, probably after we eliminated the homologous recombination capacity in the recipient cell. And because I don't think there's going to be a universal recipient cell. Nothing makes me think that's possible. So, Carol, you're up.
No, I agree with this last point. No, I wanted just to jump in to inform Jörg that here at INRAE, we're trying to do genome transplantation or to develop genome transplantation with Bacillus subtilis. For that, we're working with a group of Mathieu Jules in Paris. And so we started the project like 2 years ago or more. and actually, this is a huge challenge. And I used to say that in the yeast engineering cycle, the yeast cloning step is really easy. I was wrong. It's very easy for genome below 2 megabase. It is not for genome above 2 megabase. So frankly, we had a lot of trouble just to try to get the whole genome clone into yeast. But I hope we're getting there. And in parallel, we're trying to develop the transplantation process, working also with L-form or protoplast. And we have many, I think, obstacles to take into account. And we have, I think, many things to share with John, because I agree with him that we had to face several barriers, like the cell wall, the recombination barrier, because we have to face there are a lot of recombination in Bacillus subtilis. In Bacillus subtilis, we do not have I don't think the nuclease issue, but we're having the nuclease issue with mycoplasma. I agree, John. Even for yeast, in fact, we know now that nucleases, secreted nucleases, could be a barrier when you want to fuse mycoplasma with a yeast. We got this result lately. So nucleases are something to take into account. You're right.
Carol, do you know if there are genes in Subtilis that are toxic to yeast, like Bogomil found with the Ecoloplasma nuclease?
I believe so. I believe so, because all our attempts to clone the genome, the whole genome so far, failed. And now we have more ideas about about which elements are a problem. So we think that we have some toxic genes and also some repetitions present in the genome that create some instability in yeast.
Well, we certainly know that yeast didn't have a problem with the two ribosomal RNA operons in mycoplasma, although I suspect that if you passaged it in yeast long enough, it would have done something to that.
Maybe.
Bogomil, you seem to have a
few
question
comments
on your mind.
Just thinking of going to the first kind of topic that there is this E. coli that was split. The genomes were split into small, anywhere from 200 kb to 500 1 megabase. I think that would be another great one that you could attempt, because you can start with like ranges of chromosomes with Jason Chin, I guess, group that developed that. So that was just kind of side thing that I think would be really, I would incorporate it in the research now if you think that's possible. Now with the H-flu, so we are developing a lot of tools now for the for the RD1 strain. We are actually preparing like genetic toolbox paper. We can talk about it maybe and see how to combine this effort. But one thing, as you're thinking of the different recipient donor strain, I forgot the other H-flu that I was working. I was still at Venter Institute, but kind of like Carol said, I couldn't clone the whole genome. So I could only clone pieces of it, and there was a lot of different things that were problematic. So I still think the RD1 strain is absolutely the best as a donor and as a recipient. Mm-hmm. And the homologous recombination is absolutely fantastic for some reasons. And so we also knocking out this. So if you want to actually have probably strain without the recombination, we may have that relatively soon. So depends what you're using it for, but I would really concentrate mostly on that, because the genetic toolbox that will exist for that strain, it's from plasmid to markers to optimized methods. I think it's really kind of coming together. So and the other strain may be very problematic. We have so much problem with the other H-flu. I have report somewhere. I could even send you if I find that, but there was something. And also one thing that I noticed, if you incorporated a marker in some of the other strain and if you propagate it, I think they like spit it out and created plasmid. So you can start growing on like extremely high concentration, for example, on chromophenical, and then later when I would run the genomic DNA on the gel, you've seen smaller pieces of DNA. So they were like almost spitting something out of the genome. So again, it is a little bit data that yeah. But we did a lot of work and failed with one of the strains just for very kind of strange reasons. So just something to keep in mind. Yeah.
Yo.
This is just, I'm mostly a listener here, but John's animation of the transplantation made me think this way. But so usually, cell cycle is, G1S and what is it, G2M, and then there are checkpoint mechanisms, right? But this Capri column somehow takes in a genome from outside and think that it's replicated and start dividing. And your mitomycin result also suggests that genome broken apart is not a problem, and it keeps dividing. So maybe the checkpoint mechanism is not there in Capri column, and then maybe the cells, intra species transplantation fails because it's not a replicated genome that organisms take in. I wonder if we can engineer, skipping of checkpoint or something like that, and then increase our chance of success here.
So, Carol and Bogomil, do you think this is doable?
I think so. Just deleted a couple genes there, I guess. We just have to know what to remove. And because
we also might find that there are other yeast strains that would be perfectly comfortable taking Subtilis or E. coli plasma. Probably not, but I will have one postdoc working on this. Now they're going to have a huge advantage over what you had when you started, Carol. But they're also going to have to be, doing microfluidics work at the same time.
Mm-hmm.
So this is a hard problem.
Yeah.
But
no,
it needs
good
to be solved.
Yes, yeah, I agree. I think
I saw George Church's face light up when I said we were working on this. So
do you ever worry with microfluidics about like a lot of components getting stacked and not be able to move the fluids as you're like removing walls? Have transplantation ever been showing microfluidics? I can't remember anymore.
We never got it to work. Now, there were a lot of things that came close, and we had an experiment that it maybe could have worked, but then there was a power outage at JCVI the night that we set it up to go hours, and this just never happens. And then we found the oddity that the minimal cell looked so different in the microfluidic devices from the wild type cell, and James changed his focus to what eventually became the cell paper. It took 8 years, but anyway, James changed his focus, and so we're just getting ready to go back to this. But I have never been able to get anybody to give me money to work on this until this year. And I tried 4 or 5 times with NIH and NSF. Other questions about transplantation before we have a little free-for-all discussion? Sebastian?
Yeah, one question. There are papers in E. coli where they use an in vitro system to replicate the chromosome, and the chromosome gets supercoiled. So that really condensates the DNA. And I think that's probably a key point in transplantation, getting your DNA in the smallest volume possible so that it can enter the recipient cell. So I'm just wondering, are you considering using this in vitro amplification kit to condensate the DNA, maybe get more copies in that microfluidic chamber, and then flow the recipients in?
That's certainly something that we should consider. When Carol was here, we tried, for instance, incubating the donor genomes with bacterial protein, histone-like protein. That didn't work, and we tried some drugs that we thought might compact the DNA, and there was never any clear evidence that either of those did the job. But it's Carol's data, both in 2007 and more recently now, says that the conditions where transplantation works are extremely narrow. So as I remember, 5% polyethylene glycol was like worthless, and 10% worked. And Carol experimented to see exactly what worked. I believe that for maybe every organism, or at least every recipient cell, we may have to have experimentally determined conditions, but in a world where you can make 1 million different genomes and then test them all there, if you've got a recipient cell that you can test all of them and design, with laboratory automation, being able to produce millions of different cells using one type of recipient cell for one species, this would be a huge advance for synthetic biology, synthetic genomics.
On our side, I think, yeah, I saw that paper and I found it very interesting. And I think we will try, even during our regular transplantation process, I would like to try to check if it improves the efficiency of transplantation. And as John said, this is true that we have tried during several years to find different chemicals that could compact the DNA. Many of them did not work, but some did. I think so. But we do not have maybe the complete proof of that. But I think that some treatments that we do right now and help to improve the transplantation efficiency. The reason for this improvement come from the use of this chemical. So some of them are antibiotics that we use to prepare the agarose in the DNA in agarose plugs, like chloramphenicol, and other are. For instance, we use also hexamine cobalt, some chemicals like that. That in very old paper. The author says that they could have a role in DNA compaction. And it looks like it helps.
Something we learned when JCVI Rockville changed from the building that Carol worked in to a new building half a mile away, transplantation stopped. And at JCV, where Carol worked, we had distilled deionized water. And at the new building, they just had deionized water. Transplantation failed completely. This is something that took us a while to figure out. And even in La Jolla, to make it work, we had to go and get distilled deionized water to make it work.
Okay.
And this is the sort of thing. There was another thing that we learned about the temperature of recipient cell growth based on an incubator where the door wouldn't close properly. And so it was supposed to be It was supposed to be 37, but it wasn't. And so the conditions that transplantation work, again, are very narrow. Carol learned that sleazo grade polyethylene glycol didn't work, and that PEG 6,000 was better than 8,000. It's all, there's a lot of empiricism that has to be done here. And so certainly the postdoc that I'm trying to find, and I've had very little success with so far, is I need someone who is willing to get in and grind experimentally. But let's stop and maybe discuss as a group what are issues that we would like to address. So I will say that I've arranged with Craig, I am going to have a write-up about advances made in minimal cell and the development of this community that we will publish in PNAS. Since he's a National Academy member, you know, I can kind of grease the skids to get this into PNAS. And this will include a summary of this meeting and the one in last year. Last year with chemotherapy, I was never strong enough to write anything up, but we will do that this year and talk about what has happened in the last 6 years since the minimal cell was published. But it will offer some data about a lot of what is going on in this community. So I will get that and we'll distribute this widely to folks before we submit it to PNAS. So this is one thing we're going to do. I'm still trying to find someone in the US government who might consider changing the biosafety level of the minimal cell, but no one seems to want to, no bureaucrat seems to be willing to go out on a limb and do this. As I understand, in Germany, it's now a BSL-1 organism based on work that Sanjay Vashi did with Carol and others in INRA with a 68 gene mycoides knockout. And then there will be 2 papers that show that the minimal cell is incapable of infecting mammalian cells. One's already published last year from the Ataya group and the Osaka Women's and Children's Hospital. And then Daniela Bittencourt and I just submitted a paper to iScience that shows this in more detail. So that may help. It may even make it possible for us to send the cell to Canada. But who knows? I'm not going to drive it across the border for risk of being sent to the Canadian equivalent of Guantanamo Bay. But other issues, things that we need to do. I assume we will meet again next year, but we could meet more frequently if you guys think it would be necessary. So someone talk other than me, please. I'm hearing nothing. Silence.
Well, the meetings are really great. It's really fascinating to see what everyone is doing. And I think it's really also helping to build links and relationships between labs that had no connections before. So I would definitely really like to see these meetings become annual.
We've done that so far. Elizabeth Strahowski has suggested that I petition or put a proposal into NIH that would give me funds to run such a meeting where we actually came and were with each other. But I don't know that that would work as well.
I would. This is James here from Dresden. I think this is working really well. Sorry, I wasn't so active this time, but I also think it would be good if at some point we could maybe try to organize something where we meet in person, because that definitely takes on a different dynamic in terms of exchanging information and familiarizing ourselves with each other. Even if maybe the first time we all have to pay our own passage or something like that, it would be great to just pick a time, maybe next year or the year after, and just pick a place and go there and do this in real life.
The I'm also part of the Build a Cell Consortium, where it is aimed at trying to make synthetic cells. And we, with Kate Adamala as our leader in this, she wrote a proposal, an RCN proposal, I think, to NSF, that it actually has money, and we are able to pay at least people who are going to give a talk or a poster to come to meetings. So we have 2 of those meetings a year, but this is something that I will pursue. I did send all of my NSF, National Science Foundation program officers, invitations to the meeting, and I haven't been watching to see if any of them have shown up, because I'm trying to listen. But this is something that I am going to take a shot at to see if we could get money for this, maybe to actually have people come to visit. Other thoughts about this? It could be tacked on to a mycoplasma meeting or a build-a-cell meeting possibly, but I'm open to suggestions.
Yeah, exactly. That was my thought, John. I was thinking about what about connecting this minimal cell meeting to an IOM meeting.
Mm-hmm.
Would be great, because so many of us are interested by both communities, and I guess it could be really great.
So clearly it's too late for Osaka, but
Yeah.
But where's the next one after that? Do we know yet?
No, it's not decided yet.
Okay.
But it should be in 25.
I think so, yes.
Yeah.
Other thoughts?
Well, I'd certainly, James Dobbins-Beck, John. I would certainly keep these Zoom meetings on an annual basis at a minimum. I find them very helpful, and I always find out something new and interesting. So
you'll note that last year I had 35 talks, and this year we have 19. And I had to do a little badgering to get up to 19. Not much. I think mostly people were a little slow in getting around to sending me things, which is fine, because you guys know a guy who can add your talk even if you didn't make the deadline. But we could do this more often because it doesn't take much organization. Last year we had 90 people at the meeting. This year we've stayed around 50.
You had over 70, I think it's the first day.
And I do see a lot of names, not so much now, but when, right now there are only 21 people left, but I did see a lot of names that I don't know who they are. And I think this is super. There are more groups. Like I say, I have either have a collaboration with people, some people who I have not sent the cells to, but between that and the number of people I have sent cells to, that comes out to over 60 now. Now, some of those people may not be working with mycoplasma anymore. I know a few of those groups, like the group in Florida, that isn't doing that anymore, but still, I'm sure I will be sending out more cells to people over the next year. The material transfer agreement takes a while, not so much from our side, but usually from the people who want the cells. Take UAB, where it's taken years.
We almost have enough people for a Gordon conference.
I hadn't thought of that. But those are expensive, having just gone to my first one this summer.
Yeah, that's true.
But this format, the short talks, I think work really, really well. Everybody who gave a talk, maybe with the exception of me, did spectacularly well. I'm very pleased. I understand so much more about the cell than I did a week ago. Other thoughts? Things we need to do or not do? I'm not sending out snacks to every location. That's just never going to happen. Yo, you have any ideas?
I was just curious. So we were talking about making the minimal cell BSL1, and then Mesoplasma floram is a BSL1 organism, and I don't know, can we make SYN4 out of it, or
Well, we could, but you know,
what are the pros and cons, or what are the opinions in the field about?
if people are going to make another synthetic minimal cell, I don't think it should be a mycoplasma. You know, Clyde, I mean, ideally it would be something like a phytoplasma. I mean, a blue-green algae, like SAR11 or something. Clyde talked about wanting to make an organism. Clyde Hutchison talked to me about the idea of trying to make another minimal cell out of a organism that didn't need everything, you know, something that we could really grow in a defined medium that was more minimal. Alternatively, what would it take? What would you have to add back to a minimal cell to make it into a conventional bacterium? To make it able to do TCA cycle? Could that be done? You know, I don't want to try to build a cell wall. I think that would be really, really hard. But to make it able to look more like more conventional bacteria would be quite interesting. Jörg, do you have any thoughts about this?
Yeah, I mean, playing with metabolism, of course, is always really interesting, and I think bringing in citric acid cycle might really improve the energy, but I think it should then come also with increasing the number of ribosomal operons, because this is what accelerates growth essentially.
But that could be, I think that could be easily done. That would be the easier part of that would be a lot easier than making the TCA cycle work, I think.
Yeah, probably.
But others, James Sayans, you got any other thoughts maybe for us?
Yeah, I mean, I guess putting lipid pathways in would be would be a key step towards. Trying to turn my camera on here. Yeah, so lipid pathways would be the thing, and I mean, it's not as simple as just expressing phosphatidylcholine synthesis. Obviously, you need to have, you know, sense and response, and so I think we're maybe, you know, that would take some time to engineer. I mean, we're still trying to figure out how that works in general in any organism. So,
but I think it's easier to think about making the you know, putting in the phospholipids. You know, I don't want to have to design a cholesterol synthesis pathway.
No, yeah.
But the other things seem doable and seemed like something you could maybe guide us into doing for you.
Yeah, absolutely. I mean,
You know,
I think yeah, I mean, what we're trying to figure out now is basically dissect the minimal cells lipid regulatory pathways, which, you know, it will give us some clues about how you could potentially wire in a lipid synthesis pathway in a meaningful way where it would actually, you know, be responding to cell growth and temperature changes. Yeah, no, that would be a really fun project in the long run. Yeah.
Well, it is my expectation that over the next year I will be adding a few more bodies to work on this project. We have I was sort of off my proposal game for a while, but I'm going to be putting an ad out really soon that will bring at least one postdoc in, and I hope to get a couple more working on this system. Sebastian, any thoughts? I'm
Yeah,
picking on people.
in terms of
everybody
adding,
who's still on the call, it's coming.
yeah, so in terms of adding new functions or reintroducing metabolic pathways, my group has been working with Bernard Paulson for a few years in E. coli, but also in Mesoplasma florum. And I know Bernard is also interested in SYN3. And we've built some I-modulons. So I-modulons are groups of co-regulated genes that can work together to create a new function. And they seem to be conserved in evolution. So if you look at one I-modulon in E. coli, and you look at the same genes, they tend to be co-expressed together in other species. So we built 2 different I-modulons for amino acid biosynthesis. We've also built one pathway for arginine catabolism. And I think there are some things to do there, probably some that are more difficult than others, but I would think that this is something that will be interesting in the future. How can we start from a minimal chassis and add up to rebuild some functions?
I have been teaching the folks in Bernard's lab how to grow our cell. They bought a synthetic SP4 thinking that they could grow, or no, a commercially prepared SP4 powder that you add. And while it works for SYN1, they gave me some, it does not work for SYN3. So don't buy the thing from Remini, I think. And he's getting another manufacturer who's going to give us some to try that may work for SP4. And I will send it out to this community when we test that. We don't use FBS for anything now. We use Knockout, which is cheaper, which is cheaper. But 10x CMRL has increased in price dramatically. A 400 milliliter bottle is now about $500, at least the list price, which is not what it used to be. Shige, I see you're on the line. Have you got any thoughts for us? Hey.
Hello, nice to see you. Actually, I don't have any opinion about that, but I'm doing several experiments about using SYN3. So maybe next year I will show our recent result.
Okay.
That's it. Yeah.
Have you been able to give the cells to everybody who got permission to use them in Japan?
Yeah. Yeah.
Okay.
Many people are using. Yeah.
Good.
SYN3
I'm
cell.
really pleased.
And one guy is trying to reproduce the cell wall in SYN3, but not successful so far.
Well, so encourage your colleagues to next time talk. Should we do this a year from now or 6 months from now? What do you think?
A year is good for me, I think. And also I really welcome everyone to join the IOM Osaka next year. Yeah. Yeah.
When is that?
International Organization of Mycoplasmology, which will be held in July in Osaka, Japan. Yep.
James, I'll get you information about that.
Great. Yeah, that sounds exciting.
Anyone else? So you will all be hearing from me. Sebastian, I will, suggest a time that we can have a, intergroup meeting, intra-group meeting, whatever. But thank you all for participating. Matt Lapointe tells me that he will have the videos up soon. It'll be on the same site as before. I'm going to be posting more links to things like SynWiki and other things. Carolyn, Pascal, thank you for participating. Maria's, anyway, there are not many of us left, but I really appreciate so many of you taking, time from your Thursday and Friday. And I'm hoping that this will become an annual event, that these short talks, I think, are really, really good in that they have encouraged a lot of people who didn't know each other to know each other now, and it will improve synergy in our whole organization. Thank you very much, and have a terrific Monday.
Thanks for organizing.
Bye. Yeah,
Thanks a lot.
Thank you.
Thank you.
Bye, folks.
Bye.
Bye.
Bye, everyone. Thanks, guys.